anywhere through the recorded sessions. Then also on Auricle, notes are being provided that are basically CBME based, having CBME based question answers, MCQ, and full zone for self-assessment. And also MCQs are there from beginner level to expert level that contain standard multiple choice questions. Student can also book a slot for a personalized counseling session in which there will be a direct interaction of the student with the counselor. Then in Oracle, we have some subscription plans starting from basic to premium. Basic plan is of 3 months, standard plan is of 6 months, and premium plan is of 12 months. In premium plan, we provide services like pre-recorded classes, live class, live doubt clarification, one-to-one personalized guidance, notes, quick revision examination module, and it costs around 50,000. And also for instant 20% discount, you can use my code, that is Aanchal 20. Now proceeding with the today's topic, and also on on these various dates, we are having the class over RBC, jaundice, WBC, and platelets. Then starting with the topic, so today's we have a clinical session, that is a clinical case scenario, that is of a patient that is 60-year-old male with history of chronic obstructive pulmonary disease, present to the emergency department complaining of worsening shortness of breath and confusion over the past few days. On examination, he is found to be tachypneic with peripheral cyanosis. Arterial blood gas analysis reveal hypoxemia with a partial pressure of 55 mmHg with the patient history of COPD. Which of the following best describes the expected effect on his oxygen dissociation curve? So this is the clinical scenario asking about the status of oxygen dissociation curve. So once we will study this topic, that is of oxygen dissociation curve, so I think all the student will be able to answer. So after finishing the class, we will come to this clinical case scenario again. So now starting with today's topic, that is hemoglobin. So what is hemoglobin? So hemoglobin is a red oxygen carrying pigment present in the surface of RBC, and it contains protein, that is globin, and united with the pigment, that is heme. Okay, so what is hemoglobin? Hemoglobin is a red oxygen carrying pigment. Right? Now, talking about the structure of the hemoglobin, so we have heme is an iron-containing porphyrin called iron protoporphyrin, and porphyrin nucleus is tetrapyrrole. It consists of 4 pyrrole rings, as we can see in the structure also, that are joined by 4 methine bridges, right? Now, the pyrrole ring are numbers as 1, 2, 3, and 4. As we can see, 4 pyrrole rings are present, and also we have a central element present in the center, that is known as iron, right? So iron is in heme is the ferrous. Ferrous form means it is present as this, right? Now, the iron is attached to the n number of each pyrrole ring, and each iron combined loosely and reversibly with 1 molecule of oxygen. Then also in hemoglobin, we have a protein that is known as globin. So globin is a protein built from 4 polypeptide chain, and it has 2 alpha and 2 beta chain. As we can see, we have 1 alpha, 1 alpha and 2 alpha here, and also we have 2 chains of beta in the adult hemoglobin. Right now we are talking about the adult hemoglobin. Now we can see the heme molecule is here and globin chain is present, right? So in adult hemoglobin, when we talk about the adult hemoglobin, so we have 2 alpha and 2 beta chain, and also in adult hemoglobin, we have HbA and HbA2. So HbA is approximately 96 to 98% present, and rest 2% is HbA2. In HbA2, we have 2 alpha and 2 delta chain. Now talking about the fetal hemoglobin. So in fetal hemoglobin, we have 2 alpha and 2 gamma chain, right? So we have 2 alpha and 2 gamma chain in fetal hemoglobin, and in adult hemoglobin, majorly we have 2 alpha and 2 beta, and approximately in 2% of the adult hemoglobin, we have 2 alpha and 2 delta, right? Now we're going to discuss about some important forms of hemoglobin, right? So in forms of hemoglobin, we have oxyhemoglobin. Then second we have aminohemoglobin. Then we have reduced hemoglobin. Then fourth we have carboxyhemoglobin. And the last one is methemoglobin. All the different form of hemoglobin are very, very important, and we're going to discuss each form in very detail. Okay, so first we have oxyhemoglobin. So as the name refers, oxyhemoglobin means this is the form where oxygen can get combined with the hemoglobin. Okay, so hemoglobin react with oxygen to form oxyhemoglobin, and also it is represented as HbO2. Okay, now the affinity of hemoglobin for oxygen is influenced by pH, temperature, and concentration of 2,3-DPG also in the RBC, and a product of metabolism of glucose. Now, as the concentration of 2,3-DPG rises, the affinity of hemoglobin for oxygen falls, and the oxygen hemoglobin dissociation curve is shifted to right. So what is ODC means, what is oxygen dissociation curve, that also we're going to learn in today's class, right? Now next we have carbaminohemoglobin. This is the form where CO2 react with HP, right? So here carbon dioxide react with hemoglobin to form carbaminohemoglobin, right? Now, third form we have reduced hemoglobin, in which hemoglobin from which oxygen has been removed. That is why it is known as reduced. Now, means this is the type of hemoglobin from which oxygen has been removed, and that's why it is known as reduced hemoglobin, or it is also known as deoxygenated hemoglobin, right? Now, the fourth form we have carboxyhemoglobin, in which the HP, hemoglobin, get combined with carbon monoxide, right? Now, the last form we have methemoglobin, and in this form, when either reduced or oxygen hemoglobin is exposed to various drug or oxidizing agent, and the ferrous form means Fe2 plus is oxidized to Fe3, and the compound is known as methemoglobin, right? And it is represented as HbOH. Okay, so again I'm repeating, what is methemoglobin? When either reduced or oxygenated hemoglobin is exposed to various kind of drugs, or it is exposed to oxidizing agent. So because of this thing, this Fe2 plus get converted to Fe3 plus, and the compound together known as methemoglobin, right? So this is all about the different type of hemoglobin. Now, the different form of hemoglobin. Now we have some normal values. Let's learn some normal values. So as already we know that the normal value, it differs in male and in female. So in male, the normal range of hemoglobin lies between 14 to 18 gram per deciliter, and in female, it range from 12 to 15 gram per deciliter, right? And clinically, 14.8 gram per deciliter hemoglobin as is considered as 100% hemoglobin. right? So this is about the normal value or the normal range present in male and female. Now, what about the newborn baby? Okay, means what about the hemoglobin that is present at birth? So the level of hemoglobin at birth is very high, and the reason we're going to discuss in further classes. So at birth, the hemoglobin level is 23 gram per deciliter, and at the end of 3 months, that hemoglobin get reduced and become around 10 gram per deciliter. Now, why this kind of changes occur that we're going to learn in the class of RBC, right? So this is all about the normal values. This is all about the forms. Now we're going to learn the different function of hemoglobin. What are the different function of hemoglobin? Why the hemoglobin is considered as the very important pigment present in blood, right? So talking about the functions, we have first function that it facilitates the transport of oxygen, right? So it facilitates the transport of oxygen from lungs to the tissues, right? Now, next we have the transport of CO2. So we know that it also facilitates the transport of carbon dioxide from tissues to the lungs. So both these functions are very, very important. Then the third function we have that it also important for the acid-base balance, right? So it acts as an excellent acid-base buffer being a protein. So as we know, a very important protein present inside the hemoglobin that is known as globin. So because of globin, it act as an excellent acid-base buffer, and also it is responsible for 70% buffering power of whole blood, right? Now, the last we have that it also important for the regulation of blood flow. So it plays a very important role in the regulation of blood flow and also for BP. Hemoglobin has additional NO, nitric oxide binding site on the beta chain, which is increased by oxygen, and therefore hemoglobin binds with NO, binds with nitric oxide in the lungs and release in the tissue where it promotes as the vasodilation, right? So this is all about the different function of hemoglobin. Now we're going to learn that why hemoglobin is contained within the RBC. So we have a question that why hemoglobin is contained within RBC. Okay, and in this question also we're going to learn about the disadvantage of free hemoglobin. Okay, that what will happen if this hemoglobin is present in plasma, means it is present in free form. Okay, so if hemoglobin get dissolved in the plasma, so at that time it is known as free hemoglobin, right? If it get dissolved in plasma. Okay, so what it would lead? So it would increase the viscosity of plasma and also the viscosity of whole blood, and the BP will cause to rise, and it will also increase the osmotic pressure to the plasma and interfere with the mechanism of fluid exchange between capillaries and tissue space, right? Then second thing we have that the loss of free hemoglobin by the kidney in urine will increase, and it will lead to the damage to kidney, and also free hemoglobin is taken up and rapidly destroyed by the tissue macrophages system. So that's why this hemoglobin is present in the RBC, not present freely in the plasma. Okay. Now, another thing we're going to learn about the synthesis of hemoglobin. And again, a very important topic, synthesis of hemoglobin. So synthesis of hemoglobin requires the provision of nutrients, proteins, vitamins, mineral, especially iron, and also it take place in the developing RBC. Okay, so at what stage the hemoglobin get appeared suddenly? So it is the intermediate normoblast stage. So here we're going to learn about the factors which are responsible for controlling the hemoglobin formation. Okay, we're going to learn about the factors. So in factor, first we have the role of very important component, that is protein. So a low protein intake inside the body will decrease hemoglobin regeneration even in the absence of excess of iron present. The limiting factor here is lack of globin formation. Okay, so here the presence of protein is a very, very important factor for the formation of hemoglobin. Then next we have the role of minerals. So in mineral, first we have iron. Okay, so as we know, the iron is the main and important part, means it is a central element. So it helps in the formation of heme, and iron content of hemoglobin is 0.33%. Therefore, 100 ml of blood containing 15 gram of hemoglobin contains 15 gram of hemoglobin will contain 15 gram of hemoglobin contains approx 50 mg of iron. Okay, so we were discussing about the factors. So in factors, we have discussed about the protein part. Then we were discussing about the minerals. So in minerals, we have covered iron. Then second, we have copper. Okay, so copper is also very important for the hemoglobin formation, as it helps in the promotion of the absorption, mobilization, and utilization of the iron. And very little amount of copper is required, and it is adequate amount that occurs in diet, and most iron preparation contain traces of copper. Okay, then next we have cobalt. So again, cobalt is very, very important because it is necessary for the manufacture of vitamin B12 by the bacterial action in the lumen of GIT, and also it increases the production of a hormone that is known as erythropoietin, which stimulates the development of RBC. Then last we have calcium. Now, why this calcium is very important for the hemoglobin formation? Because it increases the iron absorption from GIT. Okay, so along with protein, minerals, we have a third substance, that is vitamins. Now, why vitamins are very, very important? Because vitamins like vitamin C, vitamin B12, and folic acid help in the synthesis of nucleic acid, which in turn required for the development of RBC, and also vitamin C. This vitamin C helps in the absorption of iron by reducing Fe3 to Fe2. Okay, so this is how all these components has a very, very important and significant role in the formation of hemoglobin. Okay, so this is all about the synthesis of hemoglobin, the components which require for the synthesis of hemoglobin. Now, next we're going to learn that hemoglobin, as we know, a protein constituting 1/3 of the RBC, and synthesis began in the pro-erythroblast, 65% at erythroblast stage and 35% at reticulocyte stage. Now, what are these stages, and what is the role of hemoglobin in these stages that we're going to learn in the class of RBC? Then in hemoglobin, we have two parts. The first is the heme, the second is the hemoglobin, globin. Then about the ontogeny of globin synthesis. So we have the time period during pregnancy they have shown. So at 3rd week of gestation, at 5 week of gestation, at 30 week of gestation, and 30 weeks of after 30 weeks of gestation, and at birth. So they have shown the various site, means at 3 to 5 week of gestation, the formation of RBC or the formation of hemoglobin will occur in yolk sac, and after these week, from 6 to 30 week, the site will be liver, and at 30 week or after 30 week, the site will be liver, and at birth, finally, the production of RBC or the production of hemoglobin will occur inside the bone marrow. Okay. Then they have shown the type of gene which is present at the 3rd week of gestation, at 5 week of gestation, like gamma and alpha, at 6 to 30 week of gestation, then on 30 week of gestation, and at birth. At birth, it will be alpha and beta. Now, hemoglobin synthesis, although heme and globin synthesis separately within developing red cell precursor, their rate of synthesis are carefully coordinated to ensure optimal efficiency of hemoglobin assembly. Now, the heme and globin produce at 2 different sites in the cells. The first is heme in mitochondria, and the second is globin in polyribosomes. Now, the protoporphyrin ring with an iron atom in center that we have learned, 4 rings are there, and with the central element that is iron. The main site is in mitochondria as it contains ALAS. Now, what is this? This is amino leucinic acid synthesis. And mature red blood cells does not contain mitochondria. So initially it will contain mitochondria, but once the RBC get matured, the mitochondria will be absent. Now, it's catalyzed by the enzyme that is delta LA synthetase. This step is the first limiting step for the whole process of heme synthesis, and it is stimulated by the presence of globin chain and inhibited by the presence of free heme group. That should be remembered, that it is stimulated by the presence of globin chain and inhibited by the presence of free heme group. Now, this represents an important control mechanism of the rate of heme synthesis, and it is coordination with globin synthesis. Several factors are required for this step, like vitamin B6, free ferrous, and copper ion. Now, the synthesis of enzymes, that is delta LA synthetase, is inhibited by the presence of free heme, and this represents a further feedback mechanism for heme synthesis. Now, about the globin synthesis. So, human normally carry 8 functional globin genes arranged in 2 duplicate gene clusters. Now, we have the beta-like cluster on the short arm of chromosome 11. So, it is present on the short arm of chromosome 11. Then, alpha-like cluster present on the short arm of chromosome 16. Now, these gene code for 6 different type of globin chains, that is alpha, beta, gamma, delta, epsilon, and zeta globin. Now, the synthesis of globin. So, as we know, it starts at the 3rd week of gestation period, and in fetal, we have alpha 2, gamma 2, and also little amount of adult hemoglobin is present, that is alpha 2, beta 2, and in adult, we have HbA, HbA2, and very little amount of HbF. Okay, so now we're going to discuss about the fetal hemoglobin. So we're going to discuss about some silent feature of fetal hemoglobin. So its structure is same as of adult hemoglobin, except that beta chain, because the beta chain are replaced by gamma chain here. Now, it contains 146 amino acid but 37 amino acid that differ from those in beta chain. Okay. Total is 146 amino acid and 37 amino acid differ from beta chain. Right? Now, third we have that it provide or it is much more resistant to the action of alkalis than HPA. Then the fourth and the very, very important one, that this fetal hemoglobin has greater affinity for oxygen. The affinity for oxygen is very nice here because of poor binding capacity of 2,3-DPG here. Okay. And also its lifespan is around 80 days. Okay. So this is all about the silent features of fetal hemoglobin. So this already we have discussed that in adult, it has alpha 2 beta 2, that is 96 to 98%, and in HbA2 1 to 2%, and little amount of that is 0.5 to 0.8% hemoglobin, fetal hemoglobin is present. Now the function of hemoglobin. So the function of hemoglobin, as we know, that it is the oxygen, it is responsible for the oxygen delivery to the tissue. And about the reaction of hemoglobin and oxygen, it is reversible. Now the oxygenation, not oxidation, less than 0.01 second required for the oxygenation. Oxygen bind to heme part and carbon dioxide bind to globin part of hemoglobin. The reaction of oxygen with hemoglobin gives sigmoid shaped curve that is known as oxygen dissociation curve. Now, one hemoglobin can bind to 4 molecules and 1 gram of hemoglobin carries 1.34 ml of oxygen at a partial pressure of 100 mmHg and carry 1.37 ml at the partial pressure of oxygen at 120 mmHg. So this statement is very, very important that one hemoglobin can bind to 4 molecule of oxygen and one hemoglobin molecule contain 4 heme group. That should be remembered. Again, I'm repeating that one hemoglobin molecule can bind or has 4 binding sites of oxygen and one hemoglobin molecule carry 4 heme group, and on each heme group one oxygen can bind easily. Okay. Now, over this statement, we have a question and I'm going to give you 10 seconds to answer that how many heme molecule are there in a hemoglobin molecule? How many heme group are present in there in a hemoglobin molecule? So we have option 1, 2, 3, 4. You have 10 seconds to answer. Yes, the answer is 4. Okay, now next question we have, each heme group of hemoglobin can bind to how many oxygen molecule? Okay, please remember here asking each heme group of hemoglobin can bind to how many oxygen molecule? Again, you have 10 seconds to answer. Yes, the answer is 1 because we have learned that in each molecule of hemoglobin, we have 4 heme group and on each heme group, 1 oxygen can bind easily. Okay. Now about the fate of hemoglobin. So as we know that the lifespan of RBC is 120 days. Now what will happen after this 120 days that we're going to learn? So at 120 days in RBC means when the lifespan of RBC will end. So at that time, no DNA, no RNA, no protein synthesis will be there. Enzymes get exhausted and there will be decreased energy production. So it increases the rigidity of cell, and also there will be triggering of cell lysis. Okay. So what will happen at the surface of RBC, hemoglobin is present. Okay. And when the lifespan of RBC get completed, so the membrane lose its rigidity and it get ruptured. And this hemoglobin comes out of the cell. Okay. Now what will happen that we're going to learn in the feat of hemoglobin. Okay. So RBC after 120 days become fragile, become weak. There will be membrane of RBC get ruptured and they are phagocytes by the reticuloendothelial system through tissue macrophages, through Kupffer cells, and by spleen. Okay. So what will happen? The hemoglobin gets splitted. Okay. The hemoglobin gets splitted in heme and globin. Okay. So this heme part and this globin part gets splitted. Okay. Now what will happen? This globin part will enter into the amino acid pool cycle and get reused again. And what about this heme part? It will also get split as iron part and straight chain of 4 pyrrole nuclei. Okay, so what we are going to learn, what will happen with this free iron. Okay, now this free iron is transported in blood with the help of enzyme that is transferrin, and also it gets stored inside the liver and get reused again for the synthesis of hemoglobin. Now what will happen with this straight chain of 4 pyrrole nuclei? This straight chain of 4 pyrrole nuclei get converted into biliverdin, a pigment that is known as biliverdin, with the help of an enzyme known as heme oxygenase. And this is very important. All the student needs to remember that this get converted into biliverdin. Then this biliverdin get reduced with the help of biliverdin reductase and get converted into free bilirubin, okay, released by macrophages into plasma. Now, with the help or with the combination with plasma albumin, it get entered in liver and kidney and also interstitial fluid. So in kidney, there is no as such action is present. Now, what will happen in liver? In liver, when this free bilirubin enters within hours, there will be absorption by the hepatic cell membrane. So as we know that hepatic cells are present inside liver, so there will be absorption of this bilirubin with the liver cells and release from albumin and it get conjugated. Means conjugation means it become water soluble. Okay. So when it get combined with gluconoric acid, so it will become bilirubin glucuronide, and with it if it get combined with sulfate, so it form bilirubin sulfate. Okay. Now what will happen? Now again, both these two type of bilirubin excreted from hepatocytes and transported through bile canaliculi uh, with the help of active transport and conjugated bilirubin present in intestine, and with the help of the bacteria, it formed the urobilinogen. Okay, so this is all about the fate of hemoglobin. And for the fate of hemoglobin, this slide is very, very important and need to remember. These two slides are very, very important, that hemoglobin gets splitted as heme and globin. Globin entered into the amino acid pool cycle. Heme gets stored inside the liver for the reuse, and these 4 pyrrole chain are responsible for the formation of bilirubin. Now, the normal serum bilirubin concentration. So total is present 0.2 to 0.1 mg per deciliter, conjugated is 0.1 to 0.4 mg per deciliter, and unconjugated is 0.2 to 0.7 mg per deciliter. Okay, this is about the normal range of serum bilirubin level. Then about the hemoglobin, oxygen hemoglobin curve or ODC. Okay, so as it name, oxygen dissociation. Okay, so it is showing the oxygen dissociation with the hemoglobin. So as we can see on the x-axis, it represents the partial pressure of oxygen from 0 to 14, and on y-axis it is showing the saturation of hemoglobin with oxygen, and the percentage is being shown. Okay, so here we can see the S-shaped curve. Okay, so now here it is representing the shift to right, and here it is representing the shift to left. Okay, so what happened in shift to right? Okay, in shift to right means there will be reduced PO2, increased PCO2, increased H plus ion, increased body temperature, and increase 2,3 DPG. Please remember. And shift to left means it is representing increase PO2, decrease PCO2, decrease H plus ion. Okay. And also this oxygen dissociation curve we're going to learn in the unit of respiration again in detail. So here it is only about the saturation of hemoglobin with oxygen or dissociation of hemoglobin with oxygen, in which here it is representing shift to left and here it is representing shift to left and shift to right. Okay. So again, the normal position of curve depend upon concentration of 2,3 DPG, H plus ion, CO2 in cell, and structure of HB. Then right shift indicates high 2,3 DPG, high H plus, high CO2, and HBS. Okay. And in left shift, it indicates low 2,3 DPG and HBF. Now, next we have a clinical condition that is known as jaundice. Over jaundice, we have a separate class after RBC, in which we're going to learn in detail about anemia and jaundice. But here we have a just little discussion about jaundice. Okay, so jaundice refers to the yellowish discoloration of skin, sclera, mucous membrane as a result of hyperbilirubinemia, means increase in the level of bilirubin inside blood when it will be above 2 mg per deciliter. Okay, and bilirubin when present more than 2 mg per deciliter, percentage for jaundice to be visible on the examination, and the symptom will be yellowish discoloration of skin, sclera, and mucous membrane. Posterior sclera first place to become jaundice. So we're going to discuss about the classification. So as the name indicates, pre-hepatic, hepatic, and post-hepatic, means before liver, inside liver, and after liver. So as we know, when the hemoglobin is not going through liver, means it is not present inside liver. So at that time, the bilirubin will be unconjugated. And inside liver, the bilirubin get conjugated. And after liver means as the bilirubin get passed through the liver, so it will become conjugated. This thing need to remember. Now what will happen in pre-hepatic condition means when the bilirubin is not present inside the liver. So at the time, the bilirubin will be unconjugated, means it is not water soluble. So this type of prehepatic jaundice can occur in the condition when there is over lysis of RBC, means more and more number of RBC are rupturing due to any condition, due to any kind of infection is present, or in the case of blood transfusion reaction or autoimmune disease, or in case of malaria. So it will cause the increase in the concentration of conjugated bilirubin. Okay. Now what will happen in intrahepatic jaundice? So as we know, the bilirubin get conjugated inside liver, so it will lead to increase in the concentration of both conjugated as well as unconjugated bilirubin, because maybe some amount of bilirubin get conjugated but some not. Okay. Now what is the cause of this intrahepatic jaundice? Due to any kind of infection which is present inside liver, or it could be due to other condition also, like in the condition of hepatitis, or in case of cirrhosis. Okay. Then what happened in post hepatic jaundice? So again, as we know that the bilirubin get conjugated inside liver, and after passing from liver, the whole bilirubin get conjugated. So again, it will lead to increase in the concentration of conjugated bilirubin and causes conjugated hyperbilirubinemia. Okay. Any causes of biliary obstruction can be the cause. Okay. Or presence of stone in the biliary duct, it can be also a cause. Then for investigation, we have liver function tests in which we test for bilirubin, the level of bilirubin. Also, we test for the various enzymes like ALT, ALP. Then also we test for the level of conjugated and unconjugated bilirubin level. Then urine bilirubin level or urobilirubin level also get tested in case of jaundice. Then, as we have discussed, in prehepatic, total bilirubin will increase, conjugated will not become normal, and unconjugated will be increased, right? Then in hepatic, total will be increased, and conjugated and unconjugated both will increase. And in case of post-hepatic, total bilirubin will increase, conjugated will increase, but the unconjugated will be found normal. Now, oxygen hemoglobin dissociation curve, as we have discussed, it signifies the oxygen carrying capacity of hemoglobin at different PO2. It is sigmoid shape that represent binding of one molecule facilitates the second molecule binding. At P50, it is the partial pressure of oxygen at which hemoglobin is half saturated with O2, that is at 26.6 mmHg. Now about the summary. So normal structure, including the proportion of globin chains, are necessary for the normal function of hemoglobin, and reduction in hemoglobin in the RBC due to any abnormality of any of its constituents result into a clinical situation known as anemia. And anemia we're going to discuss in the class after RBC. Then metabolic and other abnormality result into abnormal oxygen supply to the tissue. Clinical condition associated are anemia, jaundice, and cyanosis. So again, we have a question. How many oxygen binding sites are there in a hemoglobin molecule? And again, you have 10 seconds to answer. The options are 1, 2, 3, 4. And I know all the students now know the answer. How many oxygen binding sites are present there in a hemoglobin molecule? Yes, the answer is 4. Okay, now again, clinical case scenario we're going to discuss. So here, given the patient history of COPD, which of the following best describe the expected effect on his oxygen dissociation curve? So the answer will be shift to left. So this is all about the today's class. This is all about hemoglobin, and I hope all the students find this class more useful. So thank you so much.